Hello, everyone. Happy HD Awareness Month. Thank you for joining us for our May Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. To start, I want to thank our sponsors for this webinar, Ionis Pharmaceuticals and Wave Life Sciences. Also, a big thanks to everyone who supports the HDF. It is because of you that we are able to fund exciting science that is transforming what we know about Huntington's disease. And of course, a big thanks to all of you for being here. Today, we are going to hear from an absolute legend in the space of Huntington's disease research. We are joined by Dr. Jim Gusella, who is a Canadian molecular biologist and geneticist known for his work on Huntington's disease and other neurodegenerative disorders. His lab is currently pursuing collaborative studies at all stages of the genetic research cycle aimed at discovering genes that cause, predispose to, or modify neurological and behavioral disorders. He has had a very illustrious career in this space and was part of the Gene Hunters team that identified the gene that causes Huntington's. He's also the study lead for the GEM-HD consortium that looked at genetic data from over 4,000 people with HD and led to some really amazing discoveries like genetic modifiers that help regulate the age of symptom onset. So truly the perfect webinar guest to have during HD Awareness Month. I know I am personally very excited for today's webinar. And we are going to hear directly from Jim about his amazing career thus far. So thank you so much for joining us today to share your work. Before I pass it over to Jim, I want to remind you all that you can ask questions in the Q&A box throughout the talk. So make sure that's the Q&A box, not the chat box. We should have about 20 minutes at the end for a Q&A, so type your questions in as you're thinking of them so that you don't forget, and we will address as many of those at the end as we can. Also a reminder, this will be recorded and available on both our website and our YouTube channel in about 2 weeks. So if you want to watch it again, or if you want to share it with your friends and family, it will be available for that. So thank you again, everyone, for being here, and I will pass it over to Jim.
Thanks, Sarah. Is my slide showing up? I hope.
You're good.
All right, good. Well, this is my mandatory disclosure slide of companies that I have consulted for in the past. The only one I'm actually consulting for right now is Harness Therapeutics. But there's a lot of interest in the biotech industry for trying to get treatments for Huntington's disease, so I expect to be talking to many of them along the way. Now I got to get my slide to move, which is tricky. Okay, there we go. So this is Monte Gusella, which is a mountain that I found in northern Italy. I put up there because my name on the paternal side, the Gusella, comes from northern Italy, but I did actually not grow up there. My father moved from Italy to Canada after the First World War. After the Second World War, married a woman of French and Irish ancestry, and so I grew up in Canada's capital as a typical Canadian student, being in the capital where the government is really fairly dominant in economic activity. My parents sent me to school in French so that I would be bilingual in order to take advantage of the potential future opportunities and jobs in the area. And I grew up really as a typical Canadian kid. I was always very curious. I was interested in science and fascinated by probability and the intersection of chance and fate, which will come up a number of times in this talk. My outlook on life was actually formed by a series of about 26 books written by Walter Brooks that are about a rather what has been referred to as a Renaissance pig, Freddie, who lived on a farm in upstate New York. And this really, the books captured me and they taught me a lot of things. The things most that matched with my personality were the curiosity and the logic. But they also promoted adventure and science and friendship and community. And most particularly, they taught me a skepticism of politics and conventional wisdom. And that showed up when I finished school and went to university at the University of Ottawa, where on the first day that I arrived, they gave a values test to all of the first year students. They were doing a study of all the students, and they gave a values test where I scored off scale on the value of science on the high side, and off scale on the low side on the value of politics. And I think that has carried through the rest of my career. It was a pretty typical biology department, studying axolotls and plants and things like that. But there was one particular individual who really introduced me to something new. That was Gordon Kaplan. He had trained with the famous French biologists Jacob and Monod and introduced us to molecular biology, the study of the actual molecules that are involved in biological phenomena. He had the distinction of teaching half of his lectures in French and half in English. So we alternated day to day between French and English. And when I finished, he advised me to go to the University of Toronto to pursue molecular biology. And I went there and met David Haussman, who was actually, although at the University of Toronto, was located within the Princess Margaret Hospital, which is a cancer hospital in Toronto. And that really introduced me to the idea of doing science, not in the context of a university academic setting, but in the context of a hospital that actually was seeing patients and aimed to improve the lives of patients. And I realized that you could actually do both things at once. You could learn interesting things and you could be helpful to people at the same time. The first day I arrived in Toronto, David told me that within a year he was moving to MIT. And I had the choice of whether to go with him or to stay in Toronto and finish up the work that I was planning to start, since I had arranged what I was going to do with him in Toronto. And this is the first point at which politics intervened, which was that my wife, to be, my fiance, was in medical school in Ottawa, and we had planned for her to move to Toronto and get married. And there was a big political fight going on between the government of Ontario and the universities that allowed her to stay where she was, but not to transfer to the University of Toronto. And so I said, well, if she can't come, I guess I'll go with you at MIT. So I moved to MIT with David. His lab was focused on the development of red blood cells. It was a great lab to be in. There's this picture of the people that were there. One of whom, anybody who is an aficionado of CNBC will see that there's a TV personality there if he can be recognized. My thesis project was going to be the use of these emergent technologies of recombinant DNA to isolate the human genes for globin. These were the proteins that participate in carrying oxygen in the blood. And so I really was set on a path when I moved to MIT, and that path, as you'll see, was soon aborted. But just to get across what recombinant DNA is so that you understand what I'm talking about the rest of the time, what we knew at the time was that every human cell contained a set of chromosomes, two sets, in fact, a pair, where you got one of each chromosome from each of your parents. And essentially what a chromosome is just a very long string of DNA that is coated with proteins. And that DNA you can think of as the blueprint and operating software for the body. Everything it takes to make a person and to have that person work properly is defined in the DNA as a code, and that code allows for the production of all of the elements necessary to create the cell, have the cell grow, have the cells get together and form an individual, and have that individual operate. The code is surprisingly simple in that there's only 4 letters to it, and they pair up in a complementary fashion in the DNA. About 1% of the DNA actually codes for genes that makes proteins, and all of the rest of it is regulatory. So what we knew at the time that I started this project was that there were these 22 strings of DNA that were packaged in chromosomes, and you had 2 copies of each one. Very little of the sequence of them was known because it was not possible at that time to sequence large stretches of human DNA or of any large DNA. And so I was prepared to do recombinant DNA work, which was just evolving at that point. And what it was it enabled you to take small pieces of DNA, wherever they came from, put them into bacteria and have the bacteria copy them for you. This was called cloning. And so recombinant DNA technology really centers around cloning of DNA so that you can make large quantities of single fragments or single sequences to study them. Well, I was prepared to do that for Globin, but unfortunately, the mayor of Cambridge saw some news stories about Frankenstein-type monsters and strange orange-eyed creatures and decided that maybe this was because MIT was carrying on recombinant DNA experiments, and so the Cambridge City Council put a moratorium on them. And I was not allowed to clone human DNA for the first couple of years of my work. And even when I was allowed to do it, it was only within a very highly controlled facility that you would see nowadays used for things like COVID virus, etc. Fortunately, I was able to graduate because eventually the moratorium regulations loosened and I was allowed to clone the DNA within that P3, what was called a P3 facility. But by that time, the people who weren't in Cambridge had cloned the genes I was interested in, so I needed to come up with another project. And I developed a technique that allowed me to clone DNA that came from a particular chromosome, so that rather than looking at all of the human DNA at once, it would be possible to look at DNA from either a particular chromosome or a piece of that chromosome. And this came in handy afterwards when I needed to look at chromosome 4, as you'll see. So my plan was then to go and postdoc at Caltech with a very famous biologist who was interested in isolating antibody genes. And at that point, most of the common antibody genes had been cloned, but there was one particular form that hadn't been. And so my project was going to be to clone that. And I went out to visit California and hit two roadblocks. The lower left of the slide shows you one of them, which was that I couldn't breathe in LA when I visited there. The air was terrible. The other was that, again, with my now wife, having trained and become a pediatrician, she moved after graduating from the University of Ottawa and we got married to Boston, did her residency and was ready to go out and be a pediatrician. And it turned out that California was one of only 2 states in the country that did not accept Canadian medical training. So I flew back from California really in a quandary, wondering what I was going to do. I wasn't going to be able to move my wife to LA. I wasn't going to be able to breathe if I went to LA. And so I spent the flight, actually, honestly was in thinking about what I was going to do. I spent the flight by myself because there was a passenger in first class who was having a heart problem, and my wife, the pediatrician, volunteered to help. So she spent all of the flight in first class. And when I got tired of thinking, I actually read Freddie and the Spaceship, which I had picked up at the as a discard from the Pasadena Library. So all things come in circles. But when I got back to Boston, I was very fortunate that an alternative came up. And the alternative came as a result of Joe Martin, who was another Canadian, but he had moved from Montreal to become the head of the neurology department at Mass General Hospital. And he became familiar with me and my work through a whole series of chance events that I won't go through. But he was interested at the time in developing a center for Huntington's disease because the NIH, as a result of the lobbying efforts of Marjorie Guthrie and work, including Nancy Wexler on the congressional committee to look at Huntington's disease, had set up the idea of establishing centers and telling the NIH to fund them. And so Joe Martin decided to put in an application, and he thought it would be really good to have molecular biology as part of his center. And so he offered me the idea, the job of moving over to Mass General from MIT and setting up a lab specifically to apply molecular biology to Huntington's disease. What was known about Huntington's disease at the time was all descriptive. It was that it was an infrequent disorder and that it involved movements and other symptoms, that it onset in midlife, and that there really was no treatment. Now, that doesn't offer a lot for recombinant DNA because at the time, the only genes you could clone were ones where you knew the protein that was being made, because you could go from the sequence of the protein to try to find the unknown DNA that encoded it. But we didn't know what the gene was for Huntington's disease. So we had to have an alternative way of applying molecular biology, and that came in the genetics. The inheritance pattern of Huntington's disease was dominant, meaning that of the paired copies of the gene, you only need one to encode the problem that would ultimately cause Huntington's disease. And therefore, every child had a 50-50 chance of inheriting it. And then you could therefore, in large families, put together a track of the inheritance of that particular gene by whether the individuals got symptoms or not. Once you got people who lived a life with no symptoms and they had children, none of those children got symptoms, only the people that were carriers of the gene. So that allowed the possibility of applying recombinant DNA technology, because it turns out, as was emerging with the small amount of sequence that was being generated at the time, that about of the 3 billion bases or DNA letters in one set of human chromosomes, about 1 in 500 different between any pair. You could tell that by just looking around at people. People look different, the different hair color, different eye color, different characteristics. And a lot of that is because of small differences in the DNA. So if we could simply find differences in the DNA, we might find one that tracked within the family with Huntington's disease. And so that became the project that got me started on Huntington's disease. Was, let's try to find the location of the gene on chromosomes by finding a DNA variation that tracks in families with the inheritance of Huntington's disease. That then set up the concept of a research cycle, which I applied to Huntington's disease over the years and to a number of other diseases. But it's very straightforward. It really says that to understand a disease and to develop the means to treat it, you can use genetics. The genetics requires the study of patients' and families' DNA and combining the clinical features that say whether the individual carries a gene that causes those features and variations in the DNA that become inheritance markers to ultimately lead to the location of the gene on the chromosomes, which then you could use to identify the gene itself without knowing what the protein was. You could discover the protein by first identifying the gene. Then you could figure out what the molecular mechanism was using the more traditional techniques in molecular biology and cell biology and mouse modeling to understand the disease and use what you learned from the genetics as clues for developing an effective treatment, which you'd then deliver back as a benefit to the HD patients and families. So this was essentially the beginning of my lifelong career, applying this research cycle first to HD and in parallel to other disorders. For HD, it required families, and the families were evident both through effort and planning. Nancy Wexler, who had been part of the congressional committee that had recommended that there be centers, they had also recommended that there be a roster of families willing to participate in research. And that was set up at Indiana University under Mike Keneally. And then Nancy herself got on a plane and went to Venezuela, where there was a report of a very large concentration of Huntington's disease in a village in Venezuela. And she put together a very large pedigree of well over 15,000 individuals that both became available for this research. The first hint that the HD gene was on chromosome 4 came from a family from Midwest US in the roster, where a particular variation in the DNA traveled with the disease, although it was only a hint. It wasn't by itself statistically significant. And the first eureka moment for me came when we then looked at that sequence variation in the Venezuelan HD families And it then became clear that definitively the cause of HD was on chromosome 4. The odds were 100 million to 1 that we were wrong. So clearly the gene was on chromosome 4. So that became the time to celebrate, but it also represented the start of a new task. Life changed in essence. We now were faced with the idea of knowing where the gene was on a chromosome, but not knowing what the gene was, but we had to find the gene itself. And that changes what we had set up to do. The entire strategy became different. We now needed to be able to move along the chromosome, and we're again in the very early days of molecular biology. The identification of this marker that allowed the inheritance pattern of Huntington's disease to be followed was one of the things that helped promote the idea of a human genome project, because it pointed out that if you had the human DNA all mapped, you would be able to find things quickly when you had the markers in place. But at the time, none of the technologies necessary to move along the chromosome were available. So we set up a collaborative team that is international in scope. There are people from Germany and England and ultimately Australia and New Zealand were ultimate participants along the way. But a number of people joined in because they had different techniques that were available to move along the chromosome and try to isolate the DNA. And I'm not going to go through all of those techniques. I will point out that standing in between Nancy Wexler and Mike Keneally is Marcy McDonald, who the Hereditary Disease Foundation provided money to me after the identification of the marker to try and jumpstart this search. And Marcy was someone that I had met at the University of Ottawa, who was also advised by Gordon Kaplan to go into molecular biology. She stayed at the University of Toronto in medical biophysics and then moved to Holland. And when her partner came to postdoc at MIT with David Baltimore, I convinced her that she should come to the lab and join, the search for Huntington's disease gene. It took thousands of families, or hundreds of families at this point, becomes thousands later, from dozens of clinical sites around the world. And what had to happen is we had to compare the chromosome regions in all the families to find the exact location of the gene. And the comparison required first getting the DNA from that region of the chromosome. And since that hadn't been cloned yet, it all had to be cloned along the way and looked at. And ultimately, it took 10 years, but finally, the gene was found. Marcy and two postdocs within the molecular neurogenetics unit in her lab created Eureka moment number 2 for me, which was we now knew what the gene was, and the entire rest of that cycle then opened to try to understand how it worked and how you could develop treatments. And what the gene was something different. The variation that caused Huntington's disease was not one of these single letters that changed between chromosomes. It was actually a stretch of 3 letters in a row, CAG, that were repeated over and over again and became longer in individuals who had Huntington's disease. So this graph on the bottom right shows you the repeat lengths of CAG from individuals who have one chromosome with normal lengths and one chromosome with HD causing lengths marked by the red. The typical chromosome that causes HD is inherited with somewhere 40 to 45 repeats. Can be longer, can be shorter, but within everybody else in the population, it's also variable, but down around 15 to 25, maybe up to 30 in some cases. So everybody has this repeat, but the chromosomes that cause Huntington's disease have the repeat that is expanded and longer than would be in individuals who don't get Huntington's disease. The DNA encodes a protein, and to get there, you have to turn the DNA into RNA, and then RNA is read as 3 bases each representing an amino acid. So you go from a code of 4 letters read 3 at a time that produces a protein that can consist of 20 amino acids read off by that code. Basically, the combinations of 3 allow for 20 different amino acids. And so by having the defect identified as this repeat and finding out that it was within a gene, we were able to predict the structure of the whole gene by sequencing the region around it. And that's the Huntington's disease gene on chromosome 4. It's now referred to as HTT. It is, like other genes, broken into a whole series of pieces that are called exons. The HTT actually has 67 exons, and the CAG repeat is in the first exon. Now, all of those exons, all those pieces have to be spliced together to make the RNA, because the protein coding part is only the exons. All the rest of it is regulatory and structural. So if you think of this gene as being one of 20,000 that are in the human genome, you realize that most of the human genome doesn't actually make the protein. It regulates the production of protein from all of the different genes. The Huntington gene, I've marked out for exon 1 the particular sequence of protein that it forms in bold. And I've highlighted the sequence encoded by the CAG repeat in red, where CAG codes for the amino acid glutamine, and glutamine is represented by a Q when you're talking about proteins. And you'll notice something important, which is that at the end of the repeat of 21 CAGs is a CAA followed by another CAG. It turns out that CAA also makes glutamine. And this becomes very important later on for our studies. But for the moment, you have a picture of a very large protein. And from the point of view of what does this protein do, this sequence doesn't really tell you anything. You have to go out and do research and do studies in a variety of ways to try to figure out what it does. And I will tell you that at the time we isolated it, no one knew what it did. And as of now, no one's still 100% sure of all of the things that it does. But we put together a team within the molecular neurogenetics unit. Marcy stayed, decided to continue to work on Huntington's disease. We added Jungmin Lee, In-Seek Song, Vanessa Wheeler, who's particularly interested in instability and in fact won the Gehry Prize last year to study the various aspects of Huntington, how it might lead to Huntington's disease, what its biology was, etc. But we remain convinced that the secret to understanding the disease and the secret to understanding how to treat it lay in the genetics. And that was partly because the length of the CAG repeat showed a strong correlation with the age at neurologic onset of Huntington's disease. The longer the number of repeats you inherited, the earlier the onset of the disease, typically. But it wasn't always the case. There was variation. And you could have individuals with exactly the same CAG repeats who had onset of symptoms 20 years later than the average, or 20 years earlier than the average, or more. And when we looked at those people and we looked across the population, it was clear that the variation relative to expected onset, the change, showed some heritability, which meant that there were genes involved. Other genes beside the Huntington gene were affecting the impact of the Huntington mutation. And we refer to these as genetic modifiers. They are other genes in the genome that show normal variation, just like the kind of variation that you see that causes hair color and eye color, etc. There's a difference in these genes and in how they operate that intersects with the pathogenesis of Huntington's disease to delay or hasten onset. And so we thought, in looking at this, that this is sort of like the equivalent of what we're trying to do in developing treatments. When you try to develop a treatment, you develop an intervention that you want to modify the disease process. And if you can apply that intervention before development of the disease and it was targeted appropriately, that treatment should delay the clinical diagnosis or prevent the symptoms from ever occurring if it's a cure. Well, nature has already really performed that successful clinical trial for delaying because there are genetic modifiers that delay. And so if we could find those genetic modifiers, we would have a clue from humans to say how you delay the disease. The first thing that we learned was that 2 copies of the Huntington's disease mutation wasn't any worse than 1. There were very rare individuals who both of their parents had Huntington's disease and had the disease gene inherited in both cases. So they ended up with 2 copies, but their onset and later showed their progression is no different than having that 1 longer copy. So this argued to us that we were not looking for the gradual accumulation of damage as a result of this mutation. Something else had to happen first before the damage would ensue. And so we basically started the cycle again with, again, this belief that by studying the genetics, and you could capture with genetics the information in the individuals who showed differences in onset than compared to what would be expected, to then identify those genes and then work out how those genes work in terms of molecular mechanisms with the hope of developing a treatment. So the cycle now becomes one where modifier genes are the goal. And the technologies for looking for them have increased dramatically. When we went originally back in the early 80s to look for that first marker for Huntington's disease, we had to test individual variations in the DNA one by one. We were pretty lucky, again, chance comes in, we were pretty lucky that we found one within the first dozen to 15 markers that we looked at, but testing them one by one we knew would work eventually, but it was laborious. In the meantime, because genetics took off, because it was clear that genetics was a valuable way to approach human biology, the technologies for looking for DNA variation and for assaying it became much better. And so when we started looking for modifiers, the technology was there to look at millions of variations all at the same time. And so what this graph represents is actually taking all the variations that we look at in the human genome and laying them out on the strings from the tip of the short arm of each chromosome to the tip of the long arm. And so each chromosome is then laid out. And so chromosome number 1 shown in red, 2 in blue, and all the way across to the end of number 22. And then testing each of these variations in the DNA for saying, is it correlated with earlier than expected onset or later than expected onset? And each of these trails that goes up from the bottom and crosses that dotted line is a statistically significant location where there's a gene that is modifying Huntington's disease. And it turned out those genes fit into 2 categories. One category was a set of genes that are all part of the DNA repair or handling machinery. PMS1, MLH1, MSH3, FAN1, PMS2, LIG1, and POLD1 are all involved in handling DNA and repairing it when it's damaged. The ones in blue that showed up are not involved in those processes. And so we have 2 categories, one of which is coherent in terms of understanding mechanism. And it tied with other information that had been developed along the way, some of it by Vanessa, that the repeat showed changes in size or instability. Now, we knew when we isolated the Huntington's disease gene that when this repeat was passed on from generation to generation, it sometimes changed in size. But by studying the brains of individuals who had donated HD brain upon death, it became clear that this variation was quite extensive in the neuronal cells of the brain, where the repeats changed in size so that you might have the length of an inherited CAG repeat shown in the yellow become a much, much longer CAG repeat shown in red. And the thing is, it was happening in different cells, not all at the same time, but at variable times. You tie that to the fact that the modifiers that are involve DNA maintenance and handling and repair, and you come up with Eureka number 3, which also goes back to, remember the Huntington protein, I pointed you to that little interruption, that CAA that made a glutamine. That meant that in chromosomes with Huntington's disease, the number of glutamines is normally 2 different than the number of CAGs because you've got that interruption. Well, there are rare variants in which that interruption is lost. And by combining these 3 things together, we come up with the idea that, wait a minute, Huntington's disease is not driven in terms of its timing by the length of polyglutamine in that protein, which had been one of the major things that everybody was studying, because polyglutamine aggregates, and aggregation is involved in other neurodegenerative diseases, and probably is a factor in Huntington's disease, but it's not driving the timing of the disease. The timing of the disease instead must be driven by a property of the DNA of the repeat, and that property appears to be its predisposition to instability and expansion. That led to a 2-part model for pathogenesis. The idea that you inherit a repeat, that repeat during your lifetime can undergo changes and increase in size, and when that size, only when that size reaches a critical threshold does damage start to occur. This would explain why the people with 2 copies aren't any worse than the people with 1 copy of the mutation, because it takes one of them to reach this threshold before any damage begins to occur. So this 2 sequential component model of Huntington's disease that came from studying human population and human genetics really changed the way we looked at Huntington's disease and started to say, if you really want to get at the disease in the earliest stages, then that repeat instability becomes the important factor compared to the polyglutamine. Now, in more recent days, just this year, in fact, the technologies have yet again improved, and Steve McCarroll's lab at Harvard has added detail to this sequential model that's quite interesting. He's looked at HD brain and looked at individual single cells that remain in HD brain and found that they show evidence that the CAG repeat expansion occurs randomly and determines the lifeline of individual neuronal cells. So for most of the life of the individual, the repeat is either the inherited length or grows very, very slowly over many, many years. Once it hits a repeat length of about 80, then the rate of expansion accelerates, and it accelerates faster over a few years. But the critical thing from the point of that model I just showed you is that 150 repeat number. When it hits 150 repeats, that's when you start to see effects in the cells. You see other genes being dysregulated, you see the damage occurring, and eventually, although the repeat continues to grow, that cell is going to die. And that death doesn't take that long compared to the years that it took for the expansion to occur in the first place. But in the meantime, you've got cells that are dying. At the same time, you've got other cells that have not yet reached 80 repeats, and they're fine, or other cells that are expanding a little more rapidly but haven't reached 150 repeats, and they're fine. But once you get over that 150, the cell is fated to die, essentially. So if you look at it in terms of our 2-step model, you could plot the change in the CAG repeat length in red and see that it's measurable and it's going on in some cells, and each individual cell would have a curve that would be determined randomly by when the expansions occur. It accelerates a little bit because once you get longer, the chances that expansion occurs becomes higher. And certainly over 80, it becomes significantly higher. But the toxicity or the damage component of what's going to happen as a result of that expansion stays flat until you hit 150, and then it goes up. So if you think about what you might want to do for treatment, what you want to do is you want to come up with an intervention that stops that expansion from happening, or at least delays it. Because if you can delay that expansion significantly, then it won't make it to 150 for a long time, and then those cells remain alive and active and appropriate. So the sort of best target that comes out of the genetics right now for treatment for Huntington's disease would be to try to block the expansion that's ongoing in those neurons. And of course, the genes that we identified as part of the genetic studies point to which genes could be manipulated to try to do that. And when I say manipulated, I mean you could have developed small molecule drugs. You could use a whole variety of changes, including CRISPR type strategies, to try to interfere with those particular genes. And so this has become a major area in the biotech and pharma industry for Huntington's disease to try to interfere with the repeat expansion. And it goes well beyond Huntington's disease because there are other repeat expansion disorders. There were only a few around the time the Huntington's gene was identified in the early 90s, but the number, as the technologies have grown for looking at genes, as those technologies have improved, the number of genes involving repeats has grown and continues to grow. They're being found every week, essentially. And it turns out that in many, many of these cases, these repeats involve instability. And so the same kinds of drugs that ultimately will be developed with Huntington's disease as the target may well work in many of these other disorders as well. So just like Huntington's disease acted as a front runner for setting off the human genome work back in the early 80s with the first gene mapped to a chromosome using only the technology of inheritance with DNA markers, it continues to act as a forefront for identification of modifiers and ultimately identification of treatment for many, many disorders. In Huntington's disease, though, it's not the whole story. The clinical modifiers that we have could actually act at different points in the pathogenic process as well, because you remember that several of them were marked in blue and were not part of the DNA machinery. Now, they might act indirectly on CAG expansion, but they could also act at other parts of the process. Because the clinical manifestations that we're measuring, the things that we use, age of onset or total motor score or scoring on cognitive tests, all of these things that we can measure from individuals to test whether modifiers are having an effect, they don't result from the loss of individual nerve cells. They result from the operation of the brain and its efficiency. And that's going to be a combination of what cells are lost and how many are lost, and also the response of the circuitry and the cells around it to that loss to see whether it can make up for the deficiency. It turns out that the brain is actually pretty good at making up for deficiency because, when an individual first shows symptoms of Huntington's disease, there's already a measurable number of cells that have been lost. So up until that time, the rest of the circuitry has been covering for that loss in a way that doesn't really even allow a neurologist to immediately look at it and say, oh, you've got a problem. Only after the neurological symptoms start to appear can they say that. But meanwhile, you've already lost a number of neurons. So it behooves us to stop the expansion as early as possible, to treat the disease as early as possible. But there may be targets beyond the expansion that come out of the genetics as well. So if you think about that process, the steps at which modifiers could work are that initial rate of CAG expansion. They could shift the inflection point at which expansion rate increases. From the McCarroll studies, that inflection point is 80 CAGs, but even a shift to 85 CAGs would have an enormous impact on the age of onset and progression of the disease. So modifiers might change that. They might change the expansion rate after the inflection point because the rate is inherently different after that inflection point. Importantly, they may change the CAG length threshold at which cell damage begins. If you can move that threshold from 150 to 170 or 180, you'd again impact when the symptoms appeared and how fast they progressed. It might impact the mechanism of cell damage and loss itself. We still don't know exactly why the neurons are dying and why 150 is the critical number for causing the damage to begin. So the modifiers may act on what that mechanism is and make the cells more resistant to it. It also may simply act on the surviving cells to make them operate more efficiently given the loss of their neighbors. So there's a lot of possibilities for where these modifiers act, and we're actively studying them and trying to figure that out. If you think of it in terms of opportunities for disease-modifying treatments, the reduction or prevention of somatic CAG expansion is clearly a big target. It's one that a lot of people are working on, and they're working on it through these modifier genes, where the best targets for preventing expansion where you inhibit a protein would be this MSH3 protein or PMS1, which are both referred to as mismatch repair proteins because they are normally involved in the process of correcting mismatches where the two DNA strands don't match up perfectly with each other, because I showed you early on that they have to match up in terms of complementary A's and T's and C's and G's matching appropriately. Well, when there's a mismatch because of damage, these proteins get involved in repairing that, but they also apparently promote expansion. So you need to inhibit them in order to cut down on the expansion. There are other proteins like FAN1 that do the opposite, where FAN1 suppresses expansion. And so if you could increase the activity of FAN1, it might work. Similarly, if you just went in and reduced the length of the expanded CAG so that it never made it to 80 or 150, depending on where you go in, and CRISPR technology may allow you to do that. Or if you interrupted the CAG repeat, you would change its predisposition to expansion. So those are possibilities that are being looked at. Beyond the expansion, if you could change those thresholds, that would be great. And we're looking very carefully at whether the non-DNA maintenance modifiers do that. We don't know right now, but we're actively studying it. And third is what has been looked at for a while and continues to be looked at, which is whether the mutant Huntington is involved in the toxicity mechanism and suppressing it would help. There have been clinical trials for suppression of mutant Huntington, and those continue. There's attempts to suppress only mutant exon 1 because there's evidence that as the repeat gets longer, rather than expressing the whole protein, you sometimes produce only the protein part from exon 1. But maybe it's not even the protein. Maybe it's the RNA that's made that is causing the toxicity. And there are efforts that would also knock down the RNA. So all of these things are being studied. They all ultimately derive from the genetic understanding that has come from studying patients and their disease. And the GEM-HD is continuing this genetic analysis with even larger numbers. Sarah mentioned that we'd studied 4,000 individuals. That plot that I showed you of the variants across the entire genome that identified the modifiers actually at this point came from 12,000 individuals, and we're actively awaiting data within the next few months that will increase the number up to 27,000 individuals. And the more people you can look at, the more power you have to detect the variation. And the more genes you can find, the more opportunity you have to group them into functional processes to understand those processes and understand how to intervene. So we are continuing to try to sort that out. Probably the most important clue we have right now that we're interested in is one, because you always look at the one that doesn't fit expectation, because that's the one that teaches you something new. And that is back to that little interruption that I showed you in the CAG repeat in Huntington that goes to a CAA-CAG. There's one particular variation that eliminates that CAA. It's out there in the population at a very low frequency, but it's there, and it's on some Huntington's chromosomes. And those people who carry it typically have onset 10 years earlier than you would expect based on their CAG length. So everybody was assuming that what was going to happen was you'd look at those people and find out that the loss of that CAA in the sequence made the repeat even more prone to expansion, and the expansion would occur faster. Well, it turns out that's not true. You can look in blood, you can look in a cell model that my postdoc, Zach McLean, who talked a couple of weeks ago here, generated, or you can look in HD brain, and you can find that that particular rare variant that dramatically increases or decreases the age at onset or increases the severity of the disease, in essence, doesn't change the instability. And so it must be doing something else, and therefore it has become the most important target of our attention. While we study all the others, we really want to understand how this one works, because it's got to fit into the process. And also it's having such a big impact that if we can find a way specifically to counteract it, we probably have a big impact on all of Huntington's disease. So that's where we stand right now with the genetics. The work has been supported by lots of people. The Hereditary Disease Foundation, as I said, got us going in the early days, funded along the way with the collaborative group as well. And the Gehry Award this year has been very helpful as well. But there are a variety of other groups that have funded the work. We continue to use this paradigm of the cycle, except now we're not just identifying the causative gene, we're really focused on the modifier genes. And ultimately, it's both the patients and the clinicians who capture their information that are required for this. But I want to emphasize, if we can go back to Gusella, that this work couldn't have been done at all without the active participation of the thousands of HD individuals and families who continue to participate in the research, and we hope they will continue to participate because they are invaluable. It just wouldn't be possible without you. Thank you for your attention. Another example of genetic variation here, but I like to stop on a smiley note.
That was amazing. Thanks so much, Jim. What an amazing story. So we should have time for a few questions. I'm just going to start so that we can get through as many as we can. So this person is asking about the difference, tissue-specific differences in CAG repeat length. So why is the CAG repeat expansion from brain tissue much different than the CAG repeat from the blood, which does not usually change over time?
Yeah, so it turns out there actually is change in the blood, and in our latest GEM study, we've used that to show that the variants that cause or that modify that change in blood and the ones that do it in brain overlap but are not identical in all cases. And what it says is that the predisposition to expansion depends on the cell type. So even within the brain, it's particular cells that are prone to expansion compared to others. And it turns out that in Huntington's disease, the primary target, the striatal neurons in Huntington's, are the cells that are most prone to expansion. We do not really understand why, but it's not just because they're neurons. So for example, liver cells can be prone to expansion of the repeat. But liver cells don't cause a problem. They can turn over, and they also just don't cause the problem that happens in Huntington's. And there's some evidence from Nat Hines's group at Rockefeller that there's expansion in some neurons that don't die as a result. So there's also specificity to the death mechanisms, which again, we're hoping the genetics will sort out. So a lot of unknowns remain.
Yeah, that's great. So maybe time for one more question. So this person is asking, if there's a threshold of 150 CAGs that causes toxicity, how do you explain the finding of Peggy Shelbourne, who found neurons with repeats of over 400 and up to maybe 800?
So I said that that once you get over 150 repeats, the damage starts, but the repeat continues to expand. And you can get up to several hundred. That's occurring very quickly compared to the expansion that occurred to get you to 150. So the cells are probably alive over past 150. The cells are probably alive for a few months. And during those months, they can really expand a lot.
Thanks, Jim. So I think we're going to wrap it up there. We still have some questions, but I'll send those to you. And I want to end by echoing your sentiment. 27,000 people, that's amazing. I didn't realize the GEMHD consortium was up that high. And I think your talk did such a fantastic job of showcasing why it's so important for people to contribute to these studies and what we're learning about Huntington's, right? None of this would be possible if we didn't have this really strong collaboration that we do between HD researchers and HD families. So thank you so much for very clearly highlighting why that is so, so very important.
Well, I'm sorry I was so wordy, and if you have questions that you can send me, I will respond to them.
Thanks so much, Jim. I appreciate it. So I'm going to end by again thanking our sponsors for this webinar, which is Ionis Pharmaceuticals and Wave Life Sciences, as well as everyone who donates to the HDF to support the research that we fund. And thank you all for tuning in. And last but certainly not least, to our speaker, Jim, thank you so much for taking the time to tell us about your exciting work. Our next webinar is Tuesday, June 17th. We are going to be joined by Dr. Walter Koroshetz, who is the director of the NIH Institute that is the primary government funding source for HD research. So registration for that webinar is now open. You can head over to the HDF website right now so you can get it on your calendar. We are so proud to showcase the notable work of the HD scientists through these Research Spotlight webinars. If you think these sessions are helpful and you would like to support our mission, please consider donating today. Thank you so much for being part of our community, and I hope to see you next month.